All right, good afternoon. I'm going to finish up the Structural Immunology section of lectures that we have for Biochem Seminar this year. And we have a different emphasis this year than we did other years. But we are still going through the Wells paper, and so that's where we left off. That's where I'm going to pick up. In fact, it started with a particular question about ELISA. And that seems like a name. They use it a lot. Let's find out what ELISA is. If you look at their methods, you see there's a lot of things. You see that word ELISA right there in the middle there. After each selection, they basically they isolated 95 different clones, and they screened them. This is a lot like actually the clonal selection model. It's just being done by a robot rather than by the immune system. But what they use to detect binding, they use plates that are full of 96 well plates that are able to run 96 experiments, or 95, of ELISA that's going to detect binding. And then once it's bound, the great thing about phage is it has the DNA associated with it. So they take the DNA that made the antibody that bound the thing, and they sequence it. And then you can find out exactly where the mutations were that were selected for by the binding to the target. So they just realized that what they did is they identified 116 unique antigen binding sequences against 7 different targets. Remember that they were testing, this is how they test the 7 gene targets that they found on the volcano plots previously. So they isolated 116 antibodies against these 7 targets. And then they were going to see which antibody works best. I guess that's an average of about maybe 15 of antibodies that they find against every single one of those 7 targets. But it's important for you to know what ELISA is. This has come up in a couple of ways, but I want to have it in one place for you this one last time. It stands for Enzyme-Linked Immunosorbent Assay. And so that's where the letters come from. And what it does is it follows a primary-secondary antibody type pattern. We mentioned this before. What it uses is it uses the fact that many things will just absorb to the bottom of a 96 well plate. One particular well in it, you put your stuff in it, it absorbs to the bottom. It's those 3 colors on the bottom are the whole mess of things that you can absorb a whole lysate into there. But then you have to block the parts of the plate that aren't covered up by the lysate, and that's where the step 2 comes in. You have the little proteins that go in, non-specific proteins. Actually, commonly they're using milk proteins for this, and that's kind of why they're sort of milky colored. You're just making sure that the antibodies that come along are sticking to the thing you're interested in, not sticking to the plate. Then you put your primary antibody, which will be the thing that binds the target that you're interested in. But again, because you can't see that primary antibody, and it's even hard to label that primary antibody, it's actually easier to get a secondary antibody complex that you label that in bulk, and then you put that over, and it binds the FC region of the first antibody. Because those antibodies that you're testing will all have different V regions, they'll all have different ends of it, the fingers, but they'll all have the same feet. So you make an antibody that goes after their feet, and that antibody will do some kind of color change. The most common way to have a color change is to have an enzyme that makes color. You basically have the antibody, the secondary antibody shown in green here, is linked to an enzyme that will take an uncolored substrate and turn it into a colored substrate. And so when you see that substrate produced in that well, you know you have this entire chain of reactions. The enzyme's connected to the secondary is connected to the primary, and the primary is connected to the absorbed thing on the bottom of the well. Basically, that means that you know you have an antibody that's binding the antigen in that particular well. So ELISA shows up all over the place. There's a lot of plate readers. We're actually discussing just today among faculty what new plate reader we would like to order, because our old one is getting pretty much on its last legs. So if you use the plate reader, good news. We'll probably get a new one soon, I'm hoping. We'll have to work that out. And of course soon means in a couple of months at the very earliest. But you see how you have the whole idea. This is actually the ELISA plate there on the left. In the middle, they have a washer that will wash your stuff off the plate, and then they'll have a reader that will read the colors on the plate. You can do 96 experiments at a time. It's very high throughput kind of thing. And so you can just do a bunch of plates, and it's something a robot can do. It can pipette stuff in, it can move it around, it can wash it off, it can put it into the reader, and then we can collect all the data and use the software to put it all together. So ELISA is a big deal. If you go out and get a job in biotech, you might be doing ELISA someday. The good news is it's a lot like a protein concentration assay. It's a little more complicated, but the idea is the same. You are watching a color change, and the color change tells you that the reaction that you're wanting is going on. In this case, antigen-antibody recognition. And so antibodies are often used in ELISA assays. Antibodies are also used, you can use it in the same way to have an antibody that binds a band on a gel. What this is called a blot. Basically, you overlay. You have an SDS page gel like you have in the middle here, and then you apply an antibody to it, and the antibody produces a color only where the antibody binds. So you see, there's a whole mess of bands in the SDS page gel, but if you look over on the immunoblot, that shows you what the antibody binds, and only those 3 bands are lighting up. That shows you specifically whatever the antibody binds is specifically in lanes 1, 2, and 3 exactly at the molecular weight. This is a way for you to tell not just that your protein is the right size, but that your protein is the right sequence, the right structure. And of course, because it's big business, they have advertisements for it, and this is one of the advertisements I kind of liked. Western blot advertisements, basically blots are always like simplified gels. It's been simplified by the antibody binding the specific protein and lighting it up in some way, some color usually. And so you can have, but you really want it to be a nice tight band. You want it to be a brightly colored relative, and you don't want accidental binding to other things. That's why if you look in the middle here, it says bad antibody, and you see that there's a bunch of different bands. There's like 4 different bands. That means your antibody is recognizing different proteins. It means you probably should find another antibody because it's binding to too many things. A good blot in most cases is going to bind one single band and show you exactly where that one band is, and you see that they're advertising, if you buy our antibodies, you'll get one single band there on the right side of things. And of course they have other things that example. They have frowny bands, they have band practice, musical notes, they have felt tip markers. And I just want you to see, you can actually learn about what is a good antibody gel and an antibody marker, and a good specificity is the word I'm looking for the antibody, if you look at how these things are advertising. And so it's big business, right? So you can even go to large-scale proteomics, where basically you have a bunch of dots. Each dot's a different protein, and each dot lights up differently. And so the principle behind this might be very different on the chemical level, but the principle of reading the data means that where you see it light up, you see you have the protein that you're looking for. For example, this is something that looks like a big, huge ELISA. What it actually is proteomic screening, where they actually took, they have a serum for a U.S. adult who's never had malaria on the left. On the right, they have a malaria-infected individual from West Africa. And so, of course, the West African will have lots of antibodies to malaria proteins, right? Well, they took every single malaria protein, or at least most of them, 4,500 malaria proteins, and they put them on a chip. And then they took the serum, the antibody serum from the 2 individuals, and they put one of them over each chip. So the one on the left, you can see it hardly lights up at all. The one on the right lights up like a Christmas tree, because basically that individual has been exposed to malaria, has developed a lot of antibodies, and you can see that some of the spots light up more than others, and we wonder, are these particularly antigenic targets for malaria? And maybe this would be a good idea for us to go after these. Or maybe we should go after the ones that aren't good targets for the natural system, and maybe try to boost those. Basically, this is information, and the information is brightness equals antibody binding. It works the same for ELISA as it does for proteomics. There's one final thing that's in this paper, and I mentioned you've probably heard of CRISPR. And CRISPR is really more of a molecular biology technique than it is biochemistry, because what it does is it manipulates genes. It's like cut and paste for genes. And it's really an amazing technique. It's really fascinating to get into, and we can get into the biochemistry and the structure of it. But for the most part, when you're doing an experiment with genes, you can use the CRISPR to turn genes on and off very specifically and very easily relative to other methods. And that's exactly what they did. They sort of backed up their results by literally turning the genes on and off with a CRISPR screen. They basically knocked down, and you can do a bunch of CRISPR experiments together. They did 1,600 membrane proteins, and they knocked them down using CRISPR, and they tried to see how that fit with their results about how cell surface proteins change with cancer. The good news is they basically found that nearly all of their target proteins they found with the first screen, with the antibody screening, also showed up with the CRISPR screen. So this is just a second technique, and it didn't really have that many different, it didn't give anything that was truly unique and additive. In fact, in a sense, it took some things away because it didn't identify all the same hits that the first thing identified. But they did identify 2 genes in both experiments that came out as really strong signals. And so from the CRISPR screen, remember that they had 7 hits from the last one, and they brought it down to 5 from the non-CRISPR screen, the antibot technique. Well, this technique right here, the CRISPR screen, also gave that CDCP1, which we noticed at the end of the paper. That's what they find. It also found ITGB5. Now, they don't say much more about ITGB5. I'm not sure if they're saving it or if there's some reason why they didn't pursue it. You don't always find out in these papers. But they focused on CDCP1. It was found by both CRISPR and antibot techniques. And then you have you have the previous literature that found it for particular kinds of cancers. And now they're saying maybe it's a more general biomarker that is for a lot of kinds of cancers rather than just a couple. And that is the paper. So I have a few extra materials on this project, but I'm going to just move on now and let you finish that up. We will move on to parts 5, 6, and 7 next.